Hello and welcome everybody to my channel Omni Bioentrance, and today we will be discussing CSIR Net JRF Life Science Shift 1 question paper. Part C is being discussed here. It was conducted in July 2024. If you are new to my channel, please do like, share and subscribe. If you want still previous year question papers of CSIR being answered, you can refer my channel Omni Bioentrance. In the channel playlist, you can search for CSIR Net JRF. In that folder, you will find the previous year question papers being answered. Now coming to the next question. URA3 gene expression allows yeast cells to grow on synthetic media lacking uracil. Okay. So there is a gene URA3. It allows yeast cell to grow on media lacking uracil. Shown below are cell types having URA3 gene inserted at distinct positions on the chromosome. So one is cell type 1, it is here. Cell type 2, it is on the long arm. Cell type 2 is near the centromere. Okay. The ability of each cell type to grow on uracil, media lacking uracil in either log or stationary phase is listed on the right. So cell type 1 grows in log phase and stationary phase. Cell type 2 grows only in log phase but not in stationary phase. Cell type 3 does not grow in log phase but grow in stationary phase. The following interpretations are made, which is the correct one. Now, the location of URA3 gene in the chromosome is either in the euchromatin or heterochromatin region. That is why it is showing different types of growth in different phases. Okay. So what is euchromatin? Euchromatin, it is when the DNA is loosely packed over there. So it is accessible to transcription factors and it gets the gene gets expressed. What is heterochromatin? Heterochromatin is tightly packed DNA where the genes are silenced, and there are two types of heterochromatin. One is constitutive, which is permanently silenced, and facultative, which can switch between the active and the inactive state based on cellular conditions. So the first statement, URA3 gene in cell type 1 is probably located in the heterochromatic region. That is not true, because if it was heterochromatic, then it would not have expressed in the log phase and stationary phase. But here it is expressing in log phase and stationary phase. So it is naturally in the euchromatin region. Then URA3 gene in cell type 2 is located in facultative heterochromatin. That could be because at log phase it is expressing, but at stationary phase it is not expressing. So facultative heterochromatin, I told you, it can switch between the active and the inactive state depending on the cellular condition. So here it is based on the log phase and stationary phase. So at log phase it is switching to active form, and at stationary phase it is switching to the inactive form. So it is facultative heterochromatin. So B is correct. So wherever B comes, your answer is right. So wherever A comes, that option is not correct. Okay, B, 2 and 4 is having B. URA3 gene in cell type 2 is probably located in a chromosome region silenced in the log phase in cell type 2. URA3 is silenced in the log phase, but it becomes active in the stationary phase. In cell type 2. Wait. Cell type 2, I made a mistake there. Cell type 2 is this one. Cell type 2, it is getting active in the log phase and inactive in the stationary phase. What they have told, URA3 gene in cell type 2 is probably located in a chromosome region silenced in the log phase. It is not silenced in the log phase, it is active in the log phase. So this statement is incorrect. So wherever C comes, it is incorrect. So you got your answer already. It is 2. Now fourth one, URA3 gene in cell type 3 is heterochromatinized in the log phase. That's correct. In cell type 3, it is heterochromatin in the log phase because it is inactive there, whereas in the stationary phase it is active. So URA3 gene in cell type 3 is heterochromatinized in the log phase is correct. So D is correct. Then next one. Following statements are made regarding the nature of chromosomes in bacteria. Most bacterial chromosomes are circular, yes. However, in a few bacteria, linear chromosome exists. That is correct, as in the case of Borrelia. It is linear chromosome. All the bacteria system are known to have single chromosome. No, Vibrio cholerae is having two circular chromosomes. So B is not correct. So wherever B comes, option is wrong. So 2, 3 all wrong. So your answer is option 4, ACD. Okay. Some bacterial chromosome contain enhancer-like elements. Yes. Chromosomes in bacteria are stabilized by histone-like proteins. That is also correct. They are called as HU. So D is also correct. So ACD is the answer. Then next one. DNA polymerase Pol A has high fidelity but low processivity, and DNA polymerase Pol B has low fidelity, high processivity. Now what is this fidelity and processivity? Now high fidelity means accuracy in adding nucleotides. So it is high fidelity. So maximum mistakes are not mistakes. Maximum right nucleotides are being added. And processivity means there is a chance of the DNA polymerase enzyme to fall off the template frequently. This results in lesser DNA synthesis if it is low processivity. So low processivity means less DNA synthesis. High processivity means high DNA synthesis. Okay. Now see the question. In vitro reactions for DNA synthesis using limiting amount of Pol A and Pol B were set to further characterize the enzymes according to the following scheme. So this is tube 1, this is tube 2, you add DNA polymerase A here, DNA polymer Pol B here. Both are DNA polymerases. Okay, only thing they are named A and B. So after 20 minutes T7 DNA till 20 minutes T7 DNA is being synthesized, till 40 minutes 10 times T3 DNA is being synthesized. Here also 20 minutes T7, 40 minutes 10 times T3 DNA. Quantification of the amount of T7 and T3 DNA synthesized. Okay. So in both cases it is quantification of the amount of DNA that is synthesized. So fidelity we will not take into consideration. They have not mentioned anything regarding fidelity. They have told only amount of DNA synthesized. That is based on the processivity. You have to answer this question. Which of the following outcomes do you expect? Tube 1 will have more T3 DNA and tube 2 will have more T7. Okay, now first we will see what is happening here. Tube 1. Now Pol A is having low processivity. Fidelity we will not take into consideration because I told you it is regarding the quantification of DNA synthesized. Low processivity. So Pol A is having low processivity, so it will not synthesize much DNA in 20 minutes time scale. So T7 DNA synthesized will be very less, whereas T3 DNA synthesized will be more because it is at it takes 40 minutes. So Pol A can continue the synthesis of T3 DNA because it is at 40 minutes and it is at more concentration than the T7 DNA. Okay. So longer time will be available for DNA Pol A to synthesize T3 DNA. It is 40 minutes because it is less process. So in tube 1 T3 DNA will be more. T3 DNA will be more. Then tube 2. Tube 2 is high processivity. So 20 minutes time T7 DNA to within fraction of second it will synthesize. 10 times concentrated DNA you are providing for T3 DNA, and 40 minutes time again T3 DNA also will be synthesized. More, but the T7 DNA synthesized by tube 2 will be more than that in tube 1. Okay, that is the point. Now read the statements. Tube 1 will have more T3 DNA. Of course, I told you it is having 40 minutes time to synthesize T3 DNA, and more concentration of the T3 DNA is present than T7 DNA. So naturally T3 DNA synthesized will be more because it is low processivity. And tube 2 will have more T7 DNA. Of course, it will have more T7 DNA because in a small fraction of time it will synthesize more T7 DNA. Of course, at 40 minutes time and at more substrate concentration than T3 DNA of T3 DNA, synthesis of T3 DNA also will be more, but the amount of T7 DNA will be more than that in tube 1. So first option is correct. Now second option, tube 1 will have more T7 DNA. No. Both tubes will have more T3 DNA than T7 DNA. That is also not correct. Both tubes will have more T7 DNA than T3 DNA. That is also not correct. So first one is the answer. So next question. In order to investigate the involvement of the following proteins in mismatch repair mechanism, an in vitro reconstitution experiment was performed. A 5' nick circular DNA substrate having CC mismatch at the PST1 site was incubated with a different combination of proteins. Whereupon the repair of CC mismatch, the PST1 site will be regenerated. Okay, now this is the nicked circular DNA which is having a CC mismatch at the PST1 site. This is the repaired DNA after addition of the mismatch repair enzymes, where PST1 site is recovered or repaired. Okay. Now what they have shown here is because of the presence of the CC mismatch, PST1 site will not be able to cut this particular circular DNA. But after repair, the PST1 will be able to cut the circular DNA. Will be able to cut the circular DNA. Following the incubation, the resulting DNA were digested with PST1 and SCA1 restriction endonucleases, and the products were electrophoresed on 0.8% agarose gel. So here they have added different proteins. Some they have added, some they have not added. Okay. And after the repair mechanism, they have cut it with the restriction endonuclease, both PST1 and SCA1. So after repair mechanism, you will get a 1.1 kb band and a 1.8 kb band, or approximately 2 kb band. That is what they have shown here. It is near to 2 kb, approximately 2 kb band after repair. Now before repair, PST1 will not digest here. So only SCA1 will get digested. So you will get only a single band, that is 1.1 plus 1.8, that is 3 kb band. Now read the statements. MSH2 MSH6 complex is required for the repair of the CC mismatched DNA. If you see here, you can see that upon addition of MSH2, you get the repaired band. But same enzyme combination, without MSH2 MSH6 being added, you don't get the repaired band. So first statement is correct. Then Pol delta and MSH2 complex are necessary for the repair of the CC mismatched DNA. That is also correct. Pol delta and MSH2 MSH6 complex. Okay, here Pol delta 2 is added. 1 minute. Okay, Pol delta 2 is added here. MSH2 MSH6 complex is added. You get the repaired DNA. Whereas Pol delta MSH2 MSH6 complex are necessary for the repair of the CC mismatched DNA. Okay. Now if Pol delta is added here but MSH to MSH6 is not added, means you don't get the repair DNA. Showing that both these two enzymes are necessary for the repair of CC mismatched DNA. So 2 is also correct. Then Pol delta MSH2 complex are sufficient for the repair of CC mismatched DNA. No. See here, Pol delta MSH2 you added, you got the repair DNA. But here if you see Pol delta and MSH2 along with RAD27 you add, you get repair DNA. Here Pol delta and I forgot to take the highlighter. Laser point. Okay, so here Pol delta and XHO1 along with MSH2 you get repair DNA. Here Pol delta along with RAD27 and MSH2 MSH6 you got repair DNA. But see this lane. Here Pol delta is there, MSH2 and MSH6 is there. You didn't get repair DNA. So they are not sufficient to repair the CC mismatched DNA. So 3 is incorrect and that is your answer. That is the incorrect. They have asked for the incorrect statement. Then XO1 and RAD27 are redundant to each other for the repair of the CC mismatched DNA. So fourth option, that is also correct, because XO1 is added, RAD27 is added, not added, you get repair DNA. Here also if you see XO1 is not added but RAD27 is present, you get repair DNA. So they are redundant to each other. So fourth is also correct. But your answer is option 3. In vivo interaction between flag tagged protein and chaperone H are examined by co-immunoprecipitation. Co-chaperone A was incubated and the co-immunoprecipitation was performed in the presence or absence of cycloheximide. The result of co-immunoprecipitation experiments are shown below. Now what is this co-immunoprecipitation? So in normal precipitation what happens? You have an antigen, there is an antibody, they both bind together and they get precipitated out. But if the antigen is bound to a protein such as A, what will happen in co-immunoprecipitation is the antibody will bind to the antigen and it will pull down the antigen along with the protein A that is bound to the antigen. That is co-immunoprecipitation. So what is happening here? Now flag protein P is not added, chaperone H and co-chaperone A are added, you don't get any bands in immunoprecipitation. But when flag P is added and co-chaperone A are added, you don't get bands, only flag P protein band you get. But here if you see flag P and co-chaperone H being added, then you get flag P band as well as chaperone H band, showing that chaperone H interacts with flag P. All this is taking place, till what I told now is taking place in the absence of cycloheximide. And cycloheximide, it blocks the synthesis of new proteins. Okay, remember that point, it blocks the synthesis of new proteins. Now see this one. Now flag P along with co-chaperone H and co-chaperone A is added, you get 3 bands of co-chaperone A band also you get, showing that flag P, co-chaperone H and co-chaperone A bind to protein P interacts with H and A. But when you add all these 3 proteins in the presence of cycloheximide, you get only a flag P band but not of chaperone H and chaperone A. Okay. Now, okay, I will explain this one. Another point I have to say regarding this assay. I forgot to tell that. Okay, now cycloheximide, I told you it inhibits the synthesis of new protein, And whatever protein that you find over here, it is the pre-existing protein that is already present in the cell. That is, this condition is in vivo. So pre-existing protein that is already present in the cell is found in the band shown here in the presence of cycloheximide, because cycloheximide, it blocks the synthesis of new proteins. So when chaperone H is added and chaperone A is added, you don't get bands over there because protein synthesis is blocked. So what do you understand? Chaperone H interacts with the newly formed protein P, not whatever is present in the cell lysate. Okay, that is why you didn't get band of chaperone H over here. Now coming to co-chaperone A. Co-chaperone A, if you see that co-chaperone A and flag P protein you add, you get only flag P protein, but you don't get the bands of co-chaperone A here, showing that co-chaperone A doesn't directly bind to flag P protein. But in the presence of chaperone H, co-chaperone A binds, showing that co-chaperone A binds to chaperone H, which binds to flag P protein. Okay, so that is what is being depicted in the diagram here. Now coming to the statements. Again you have to see for the incorrect statement. Protein P interacts with chaperone H and co-chaperone A. Of course, when both are present and protein P is not there, there is no band. Okay, so one is correct. Chaperone H interacts only with the newly synthesized protein P. I told you when cycloheximide is added. So that is also correct. Interaction between co-chaperone A and protein P is independent of chaperone H. No. It is incorrect. Because co-chaperone A has to bind first, then only co-chaperone H has to bind first, then only co-chaperone A will bind. So interaction between co-chaperone A and protein P is independent of chaperone H. Is a wrong statement, but that is the incorrect statement, so that is the right answer. Then fourth one, interaction between chaperone H and protein P is independent of co-chaperone A. That is also correct. When co-chaperone A is not added, that is this one, you get band of co-chaperone H and flag P protein. In lane 3. Now next one. Given below are few statements related to enzymes and their functions in molecular reactions. Alkaline phosphatase remove 3' phosphate from DNA and RNA. That is incorrect statement. You have to see for combination of all correct. So A itself is wrong. It removes the 5' phosphate from DNA but not 3' phosphate. So wherever A comes in the option, that is wrong. So 1 and 4 wrong. S1 nuclease removes single-stranded regions from partially double-stranded DNA. That is correct. It is a single-strand specific nuclease. So B is correct. Okay, B is there in 2 and 3. 5' end labeling DNA molecules can be done by using polynucleotide kinase, which transfers the P32 labeled phosphate group to the 5' end of the dephosphorylated DNA. That is correct. So that gives your answer as B and C. Option 2 is the right answer. 3', 5' exonuclease activity of Taq polymerase. Taq polymerase has 5', 3' exonuclease activity, not 3', 5' exonuclease activity. So D is wrong. Then next one. The ratio of total protein content to total RNA content was measured in yeast cells in the log phase during growth in minimal media and in minimal media supplemented with amino acid cocktail. Okay, the total protein content and RNA content was measured in yeast growing in minimal media and minimal media supplemented with amino acid. In the latter case, that is when amino acid was supplied, the ratio of protein to RNA increased dramatically. Which one of the following is a correct inference of the above information? In minimal media, proteins are degraded at higher rate. There is no evidence in the question that they are degraded at higher rate. They have not mentioned anything like that. So first is wrong. In amino acid supplemented culture condition, fewer ribosomes are simultaneously active on a single transcript. That is also not correct. If fewer ribosomes were active in amino acid supplemented condition, then it would have reduced the translation and would result in lower protein to RNA ratio. Okay, so 2 is incorrect. Then in minimal media, amino acid availability limits protein translation to a greater extent than transcription. That is correct. In minimal medium, amino acid availability, that is in the first condition, that is this minimal medium, not the amino acid supplemented medium, limits protein translation to a greater extent than transcription. Yes, because in this minimal medium, amino acid is not being added. So amino acid availability is limited. So this lowers the translation, protein translation, because ribosomes have got fewer access to that particular amino acids. So translation will be less, but transcription will be greater, because transcription doesn't depend on amino acid content. Okay, transcription is DNA to RNA conversion. So it doesn't depend on amino acid conversion. So the transcription will be greater and 3 is the right answer. Amino acid supplementation reduces RNA synthesis by modification of RNA pol 2. There is no indication in the question that RNA pol 2 is modified, etc., etc. So 4th is not correct. Then next one. SLN1 receptor, a part of 2-component system, is required for osmoregulation in yeast. Now forget about 2-component system. Just remember SLN1 is required for osmoregulation. Yeast mutant, that is SLN1 lacking, the SLN1 receptor protein dies. So a researcher, what he does is he tries to rescue the mutant by expressing an Arabidopsis gene for the cytokinin receptor CRE1, which like the SLN1 is a histidine kinase that acts through 2-component system. Okay, now SLN1 receptor is required for osmoregulation in yeast. Yeast mutant that is lacking the SLN1 receptor, it will die. Now the researcher, what he or she wants is, he wants to rescue the mutant. And for that what he does is, he expresses the cytokinin receptor CRE1 from Arabidopsis. Why? Because CRE1 and SLN1, they both are histidine kinase and act through 2-component system. So will he be able to rescue it or not is the question. The following statements are made regarding the outcome of the experiment. First one. SLN1 and CRE1 protein respond to the same external signal, therefore CRE1 rescues. How do they respond to same external signal? SLN1 doesn't respond to cytokinin. CRE1 respond to cytokinin. So that is not correct. They don't respond to the same signal. It is different signal, but they are they both are histidine kinase that act through two component system. So not the external signals are same. That will not result in rescue of the yeast mutant. As CRE1 cannot interact with the downstream signaling pathway in the yeast, it will not rescue the yeast mutant. That is also incorrect. Now it could be possible that CRE1 to rescue the mutant if the domains are compatible. Okay. If the intracellular domains are compatible, not the extracellular. Extracellular domains are where your ligand will bind to the receptor. But if intracellular domains which stimulate the downstream signaling, if they are similar, then they can rescue the yeast mutant. But here they say it cannot rescue the yeast mutant, so B is incorrect. CRE1 will rescue the yeast mutant only if cytokinin is present. Yes, because CRE1 it binds to cytokinin, it is given in the question. Then the effector domains of CRE1 and SL1 protein are sufficiently similar, and therefore CRE1 can induce the downstream signaling pathway and rescue the yeast mutant. This is what I told just now. If the domains are similar, that is your intracellular domain or your effector domains, they are similar, then CRE1 can induce the downstream signaling pathway and rescue the yeast mutant. So C and D are correct. Answer here is option 2. Then next one. A researcher was interested in detecting parasite-derived antigens in Plasmodium falciparum infected erythrocyte. Parasite-derived antigens. The following labeling experiments were performed, followed by immunoprecipitation with antibodies against falciparum proteins. And autoradiography. Then labeling with P32 ATP in media. That will not give Plasmodium falciparum proteins. Labeling with iodine 125 in the media. That will also not give falciparum proteins. S35 methionine in the media. That will give, because it incorporates into protein during translation. Okay, so C will give. D also will not give because it is protein. Now you will be doubting whether erythrocyte is also having proteins. No, because erythrocyte doesn't have organelles to synthesize proteins. Now S35 methionine, I told you it will get incorporated during translation. So it will label only the falciparum proteins, not the erythrocyte proteins. Okay, S35 methionine. Erythrocyte doesn't have a translation machinery because it doesn't, I mean a mature erythrocyte. Okay, because it lacks organelles such as nuclear, ribosome, mitochondria, then what, endoplasmic reticulum. So all these organelles are necessary for protein synthesis, but it is lacking in erythrocyte. So C is the right answer. Then next one. Overexpression of protein A in the brain of Drosophila melanogaster causes the degradation of ovaries in the animal. Now there is a protein A in Drosophila. Now if it is overexpressed, that is you increase the amount of protein A, it will cause degradation of ovaries. Now overexpression of a secretion incompetent allele of A does not cause this phenotype. Okay. However, down regulation of protein B in the ovaries. Okay. Now there is a protein B in ovaries concomitant with overexpression of protein A. Now if you down regulate protein B in the ovaries and you overexpress the protein A, in the brain, it prevents ovary degradation. That means there is some connection between B and A. So A and B are found to physically interact in the ovary lysates. So that is the point here that you have to remember. B in ovaries with concomitant overexpression of protein A prevents ovary degradation, and they physically interact in the ovary lysate. A and B are found to physically interact in the ovary lysate. In the light of the above experiments, which of the following inferences would be correct? The protein A cell autonomously influences ovary development, while B is secreted to influence brain function. A needs to be secreted from the brain to affect the ovaries. There is no indication that B influences brain function. So A is not correct. First is not correct. A is a ligand secreted from the brain and B is a receptor in the ovaries. That matches here. Which one? Which I have underlined. B in ovaries, down regulation of protein B in ovaries with overexpression of protein A prevents ovary degradation. That means there is no receptor for protein A to, there is no receptor for protein A to bind. Okay, that is why there is prevention of ovary degradation. So A is the ligand secreted from the brain and B is the receptor in the ovaries is the answer. Now third one. A is a neurotransmitter secreted from the brain and B is the signal transducer in the ovaries. Now neurotransmitter, they affect shorter distances, not large distances. Okay. So brain to ovary is a very long distance. So 3 is not correct. A cannot be a neurotransmitter. A is a receptor secreted from the ovaries. A is secreted from the brain. So fourth option is not correct.